All right, let's talk about the tiny microscopic engines that basically make you, well, you. We're talking about enzymes. And trust me, without these things, you literally wouldn't be here.
Have you ever just stopped and thought about what's going on inside your body right now? Trillions, and I mean trillions of chemical reactions are happening every single second. Digesting food, copying DNA, sending signals, it's like this massive, non-stop chemical factory. So how on earth does it all happen so fast and so perfectly without, everything just grinding to a halt?
Well, here's the crazy part. If you left those reactions to happen on their own, without any help, most of them would take years. Some would even take centuries. Life just doesn't have that kind of time. It simply couldn't exist at that pace.
So what's the secret sauce? What's the cheat code? It's this number, 10 million times faster. Enzymes are the ultimate accelerators. They can speed up reactions by millions, sometimes even billions of times. They take something that should take a lifetime and make it happen in the blink of an eye. It's just incredible.
So what is an enzyme really? Think of it as a biological catalyst. Its whole job is to make chemical reactions happen easier and faster. Every reaction needs a little push to get started. That's called its activation energy. You can picture it like a big hill you have to climb. What enzymes do is they basically build a tunnel through that hill. It makes the journey so much easier.
So first, let's really dig into why these things are life's indispensable accelerators.
Okay, so how does an enzyme pull this off? What's the trick? Well, it all boils down to one very special spot on the enzyme.
This spot is called the active site. It's this unique little 3D pocket that's perfectly shaped to fit its target molecule, which we call a substrate. The classic analogy, and it's a good one, is a lock and key. The substrate is the key, and the enzyme's active site is the lock. Only the right key will fit. But it's actually even cooler than a rigid lock and key. It's more like a glove fitting onto a hand. When the substrate binds, the enzyme actually changes its shape a little bit to create a perfect, snug grip. We call this induced fit. Think of it like a firm handshake. That little squeeze makes all the difference.
So let's walk through the whole four-step dance. Step 1, the substrate binds to the active site. Step 2, we get that induced fit, that perfect handshake, forming what's called an enzyme-substrate complex. Step 3, this is where the magic happens. Catalysis. The enzyme puts just the right amount of stress on the substrate, lowering that energy barrier. And finally, step 4, poof, the reaction's done, new products are formed, and they're released. The enzyme itself is totally unchanged, ready to go again. It's a perfect little recycling machine.
All right, so we've seen how one little enzyme worker does its job. Now let's zoom out and look at the whole factory floor, because your cells are basically these bustling factories, and the enzymes are the entire workforce. And just like in a real factory, they're not all doing the same thing. They're split up into different departments, each with a very specific job. And believe it or not, biologists have actually organized them into 6 major departments, or classes. You've got oxidoreductases. They're like the power plant crew, managing electron transfers. Then there are transferases, moving chemical groups around like an assembly line. Hydrolases are the demolition team, using water to break molecules apart, super important for digestion. Liases also break things apart, but they do it without water. Isomerases are the interior designers, just rearranging the atoms inside a molecule. And finally, you've got ligases, the construction crew, gluing molecules together. Think DNA repair? That's their gig. Now, you can't have a factory with thousands of workers just doing whatever they want, whenever they want. That would be chaos, right? The cell needs a management system, a way to tell enzymes when to work and when to take a break. So let's check out how it switches them on and off. One of the main ways to control enzymes is with inhibitors, basically molecules that tell them to stop. There are 2 main kinds. First, you have competitive inhibitors. I like to call these the imposters. They look so much like the real substrate that they can fit into the active site and just block it. It's like someone parking in your designated spot. Then you have non-competitive inhibitors, or the shapeshifters. These guys don't even try to bind to the active site. Instead, they bind somewhere else on the enzyme, and that binding changes the shape of the active site. So now the real key doesn't fit in the lock anymore. Super clever, right?
Here's another brilliant strategy. Some enzymes are built with the safety on. They're produced in an inactive form called a zymogen. Think of a powerful tool that's shipped with a safety lock in place. It only gets activated by having a tiny piece of it snipped off at the exact time and place it's needed. This is super important for, say, digestive enzymes. You don't want them chewing up the very cells that made them before they even get to your stomach.
And for even faster on-the-fly control, the cell has another trick up its sleeve. It can stick on or pull off little chemical tags like a phosphate group. This is called covalent modification, and it's basically a light switch. Flip it one way, the enzyme is on. Flip it the other, it's off. This allows the cell to respond to its needs in real time. It's an absolutely beautiful and dynamic system.
Okay, so what? Why is all this microscopic factory stuff important? Well, because this isn't just some abstract concept from a biology textbook. This is what's keeping you alive. It's happening inside of you and all around you this very second.
Let's connect the dots. Remember those hydrolyses? They're in your gut right now, breaking down the food you ate. Those ligases? They're buzzing around your DNA, constantly fixing little typos to keep your genetic code stable. And it goes way beyond just our bodies. A huge number of medicines are designed to be enzyme inhibitors. Penicillin, for example, works by shutting down a key enzyme that bacteria need to live. Your liver is packed with cytochrome P450 enzymes that detoxify your blood. Heck, we even put enzymes in laundry detergent to literally eat the stains off your clothes. They are everywhere. And that brings us to a really exciting place. We're getting so good at understanding these tiny engines that we can actually start designing our own. So that leaves me with one final question for you. Knowing how powerful and precise these things are, what's the next big problem we could solve by building the perfect enzyme? Could we design one to break down plastic in the ocean, or maybe target a cancer cell? The possibilities, really, are just about endless.